This is TWIS. This Week in Science, episode number 899, recorded on Wednesday, November 2nd, 2022. Pick your nose or snot? Hey everyone, I'm Dr. Kiki, and tonight on the show, we are going to fill your head with time, trees, and lots of nose picking. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. The more you learn about anything, the more you know. The more you know, the more you understand that thing you learned about. But more importantly, what you know about shapes how you understand the world around you. And there's nothing in the world more well-rounded than an understanding of science. Be it animal, mineral, or vegetable, from the complex to the seemingly obvious, from quantum to galactic in scale, from cutting-edge innovations to the most ancient of mysteries, science has you covered. And there's no better way to get an understanding of science than learning what science is happening now, which happens to be the theme of This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again. And yes, like I said, there's going to be nose picking on the show tonight among...
What do you keep going on about?
It's science,
well,
Justin.
I don't plan on actually picking my nose tonight. I don't know about you, Kiki, but we are definitely going to talk about it.
We are going
I think if we actually do it, we might be able to boost some of the viewership.
I'm not so sure about that, but I'm not willing to do this experiment
TikTok
live.
Is that how it works? The TikTok all the kids are into?
I guess so. I don't know. Too old, but not too old for the science. We've got all the fresh science tonight. I have stories about, what do I have? Quantum time, protein prediction, nose picking, like I said, and vomiting. Because you got to round it all out.
Where were you last week? This sounds icky and spooky and gory and gross.
I know, there was even a Black Widow memory story, and I was like, oh man. Yeah. But we won't talk about that tonight. That's just a tease because it's not Halloween anymore, although we would sometimes prefer Halloween every day. Anyway, Justin, what'd you bring for the show?
I have baby zebrafish getting their brains pruned by microorganisms. DNA from some ancient humans in South America. There's kind of a little, maybe a mystery there. Why Americans are dying younger than other people in the world who have similar type economies. And and a look at some of just a fun look at how many trees have been proposed to be planted based on carbon offsets.
Spoiler,
so many.
not enough.
Well, you'd be surprised, actually. They got very ambitious with this one.
All right.
Yeah.
Tree ambition. Don't prune back. Okay, Blair, what's in the animal corner?
Oh, I have another story about nose pickers, but these are animals.
Okay.
Non-human animals, specifically. And then I also have a study about playful bees and mealworms, the new marshmallows.
Nope.
Excuse me? No.
Oh, yeah.
Didn't like the old marshmallows either. Didn't like the old marshmallows. I don't think I might like the new ones any better.
Mealworm flavor marshmallows now at a Stay Puffed near you. All right. Well, this is, as I said, This Week in Science. And if you are wanting to follow all the insanity of the science of this show, you can find us all places podcasts are found. We also stream weekly. No, not with the mealworms at the disco. We stream weekly on YouTube, Facebook, and Twitch, 8 p.m. Pacific time on Wednesdays. And you can find us as a TWIS Science if you look for us on Twitch, Twitter, and Instagram, everywhere else, TWIS or This Week in Science. And our website is twis.org. We would love your likes, your follows, and anything else you can do to help us go up those algorithm rankings. But now it's time for the science. Time. How do you count it? How do we do it? Time, right? You count a second. Oh, Blair's got her watch up, right?
In cups of coffee?
Well, that actually could be a more absolute way to measure instances of time. But really, when you're talking about time in the quantum realm, how do we do it? Time in biological space-time is linear, right? We're looking at 1001, 1002. Yes, we go one way. It's very relative, right? It's just like this 1 second, and it's time 0. And if you're talking about science, we always have, we started the experiment at t equals 0, and then t equals 1, t equals 2, you take your measurements at these various time points. But the smaller you get, and the more into the atomic, subatomic, quantum frame you go, it becomes wishy-washy, a little different. You can't count things as exactly because they're such small integrals of time. And additionally, not just the absolute smallness of it, is you throw probability in there. And you've got things winking in and out of existence. You've got points, you've got waves, you've got not time as the way that our biological brains consider it. So really at the quantum level of things, time doesn't really exist, but we still need to count it somehow. And so researchers...
Which is time travel work if you go to the quantum realm, right? That's how it's...
Yeah.
That's how they explained it in the superhero movies. Got it. Okay. I'm on board.
Yes! You're following us. Thanks to the Marvel universe for helping with this.
Yes.
Doctor Who and the Marvel universe. That's right.
Yeah.
That's right. Anyway, in the latest physical review letters, researchers from the University of Uppsala in Sweden have written up their new way of measuring time, and it all has to do with electron fields in atoms. Now, atoms, the way that we used to learn about them, you'd have the little nucleus, proton, neutron, surrounded by rings of electrons. Well, now we know those rings are not just single or whatever electrons going on. It's clouds of probability. And so if we hit those electrons with laser beams, pulses of light, we can energize them, and the clouds puff up because the electrons get energized, and they're like, I want to go to higher levels. And so they get puffed up. And so it's like expanding electron clouds. And this is called a Rydberg state after the people. That's the mathematical way that they are quantized in this particular fluffy wave of energetic wave packets. Now we found out, though, previously, that if you energize one electron cloud and you make another electron cloud and they're kind of near each other, just like ripples on a pond, they can interfere with each other. And so the interference patterns in themselves are unique. They have unique fingerprints, and the researchers suggest that if you have these multiple wave packets of these fluffy Rydberg clouds bouncing into each other and rippling against each other in the quantum pondiness, then what you have is a particular fingerprint that can be used as a measure of absolute time, not related to any clock, but that can be used as these interference patterns dissipate and change as the energetic levels of these electron clouds change. They can excite these atoms and allow their measurements for up to 81 picoseconds, which is really not very long.
That's a measurement of time already.
It is a measurement of time.
TikTok time.
It is a measure of TikTok time, but you don't have to start at time equals 0. It can be whenever it is. The measurement, if you're looking at a system potentially that you don't know the instant initiating state of, you don't have a time equals 0, you can look at the interference patterns and go, oh, that's unique. We look at that pattern and how it changes, and suddenly you're able to measure time at the quantum level. So what they're doing is they're creating a quantum watch, or as they call it.
But if it doesn't start at 0 and it doesn't connect it. So, hey, everybody come over to my house. I'm throwing a party. When is it? It's going to be at the time it happens, and that's it.
Yes.
That's not fair.
You get it. You totally get it. That's exactly it. Yes. So these energetic wave packets, these Rydberg packets, they're having parties that you just have to, come across. But the idea is that we will now be able, with the accuracy that they're able to measure, 81 picoseconds with an error rate of like 8 femtoseconds. I mean, we're talking like 10 to the minus 18 here. We are getting at very, very small error levels, which means high accuracy, which means that we can start to really get rid of error rates in quantum computing. We can start developing measurement systems for smaller and smaller technological states as we try and advance our technology into more of these subatomic levels. So, yeah. Time is everything. Time means nothing at this stage, but we have to know how it works. We have to have some sense of measurement in a probabilistic environment. It's, mind-bending or watch turning, but it is what it is. Yes. Justin, you want to tell me a story?
Oh, gosh, I would love to. We're going to start off with the baby zebrafish.
Okay, let's go with some baby. I love baby zebras.
Baby zebrafish, which I guess they call them larvae?
Larval fish.
Larval baby zebrafish need microorganisms in order to make friends. But it's all in their head, apparently. Published in PLOS Biology, University of Oregon researchers looked into the neuronal and social development of zebrafish larvae with or without their normal microbiota for the first 7 days of development. They looked at them at day 14, and they found that social behavior at day 14 was inhibited in the group that was denied their normal microbiota for just that first 7 days, compared to their siblings that had the normal start of microbiota. So the brains, then, of the restricted group, when they looked there, had fewer microglia immune cells in their forebrains. They had denser, more complex neural branching patterns and lower levels of an active gene involved in pruning neural connections. So genetically reducing the microglia without affecting the microbiota, so they had a second round here, they got the same results. They had this lack of pruning that was taking place, this complex branching, and fewer microglial cells. So normal neural and social development was restored in these germ-free larva baby zebrafish by adding fishy probiotics back to them, indicating that the pruning activity is actually sensitive to or dependent on gut microbiota. Studies first demonstrate that the microglia are required for pruning of neural connections in zebrafish larvae, and that an intact microbiota is essential for that normal pruning and then later fish social behavior to take place. So somehow, symbiotic bacteria gut encouraging social behavior by promoting the ability of pruning in the brains of zebrafish manipulating them into being a more social creature.
So now my question is, like you just said, manipulating. So is this, is vertebrate social behavior all just manipulation from microbes? Microbes want to expand around the globe more, and so they have pushed our nervous systems to become more social, so we interact with each other more. Mm-hmm. So, I mean, I'm expanding this from fish, but
we're the city bus. This is what I always say. We're the city bus. They're taking us to their next destination, and they're telling us where they want to go with, all of these chemical signals and stuff like that.
The developing gut-brain connection here.
Oh, this is, and we're learning so much about just that gut-brain connection in an ongoing fashion. This is fascinating.
It's also, I think it's also interesting. So I'm not really sure how the pruning makes one more social. I don't get this aspect of it. Maybe you can explain to me, Kiki, a little bit if you've got it off the top there. What is this pruning? It sounds like you're inhibiting brain development, but on the other hand, it sounds like you're preventing it from cascading into something that's not socially cohesive.
Right. So every, the mammalian brain, fish brains, the human baby brains, lots and lots of neurons to start off with, because you want as much input as possible, but the pruning itself is what ends up basically just tuning in particular patterns, particular pathways, and then particular behaviors. And there's a study out this week also that we used to consider that in the autistic brain, it was only neural pathways related to like social behaviors and emotions, but they're finding that it's much further distributed throughout the brain. There are pathways that have been affected throughout the autistic brain during development. And so this from the fish brain perspective is also very interesting, because what's happening is you have the gut influence that is leading the brain to basically fuse in certain pathways for survival, for that social behavior that's going to allow them to potentially stay within the school of fish that's going to protect them, to potentially stay within that school of fish that's going to lead to reproduction later in life, to stay within that school of fish that's going to lead them to the food source that they need. So the social behaviors are, it's you don't want too many neuronal inputs and outputs, because that is more chaotic than having it pruned.
Okay. I just now want to know what happens if you I guess you can't not have a microbiota at some point, because you'll be ill for other reasons. So they have to give, you can't run the experiment forward and just see what happens to the brain, because it's going to be a cascade of other problems.
You do it in the fish. You do this in the zebrafish, and you see what happens, and you find, oh, they're not social. And what happens when zebrafish are not social? You have these other issues. And so, But you don't do it with people. You do it with fish.
And they only did it for 7 days and then restored it. I'm wondering why they restored it. And I'm sort of thinking around the part that wasn't in the publication about what happened to the fish if they didn't restore it. Maybe they were just like, they didn't develop at all. I don't know. I need to see the part of the study they left out. It wasn't, we get it.
What happens after those 7 days? What about Bob the fish who didn't make it? Yeah. There's always a control group. So, there are fish for sure that they probably did not
Oh
restore
yeah,
the
there were.
microbiota
So
too.
it was in
and
comparison. It was in comparison to their control group that had the microbiota all the way through. That was there, but I didn't see the extra anomaly group, uncontrolled group, where they didn't allow them any microbiota beyond that 7 days. That's why I'm suspicious that something else was a problem at that point, because that would be a nice third category to see. But
yeah, it could have also been it was an output. This is their one of their first experiments into it, and so they decided to have a time output because you put limits on your experiments, right? You say, this is the experiment I'm going to do. We're going to do it for this long. We're going to sacrifice this number of fish. We're going to look at their brains. We're going to look at their behavior. This is what we have to work with. Or you have a particular amount of time in the lab that's going to only pay for a graduate student or a lab assistant to feed the fish and take care of the water for the fish for a period of time. So everything's done within constraints. So potentially this is the beginning, and maybe there will be those further studies, or this is what they published so far and they haven't published further than that. I don't know, but it's a
regardless, great correlation.
Yeah,
I want to know. I want to know this mechanism. How is our brains controlling our social interactions through brain pruning?
No.
Could that be a thing?
Totally, yes, it is a thing for sure in humans. But the big question is, how is the microbiota influencing it through our gut during our development?
Right.
Because from fish, immediate next step is to what's going on with us. Blair.
Yes.
I know you don't want to talk about us.
No.
You want to talk about
eyes. Yes. Eyes. AYE-AYE.
This is that. What kind of creature? All I can picture is that long finger.
Talipo. Give me back my Talipo, if you're familiar with that.
No. What?
Anyway, no?
Neither of us have any idea what you talk about.
Okay, we'll Google that later. It's very good for Halloween, actually. No, eyes are nocturnal lemurs. So they're prosimians. They're related to monkeys, but they are kind of pre-monkeys evolutionarily. All of the lemurs on Madagascar, the only place lemurs are found, they are an evolutionary precursor to monkeys and didn't have selective pressure like monkeys on the or prosimians on the mainland did. And so they have these kind of like dog-like faces. They're not as smart as monkeys. They don't have a lot of the things that monkeys that simians have, because on the island of Madagascar, they didn't have the same selective pressures as the same predators, right? So anyway, eyes as a nocturnal lemur are famous for having this very, very long middle finger, 8 centimeters long. So if you picture an animal this big, then they have, there's about 2.5 inches or centimeters to an inch. So you're talking 3.5 inch long finger. So like a finger as long as my finger, when the rest of their fingers are like a couple knuckles, right? So much longer than all the other fingers. And they also are really thin, and they usually use these to get grubs and things out of trees. So they'll tap, 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 find where it's hollow, and then they have specialized teeth to scrape down. You can actually scrape down cement when you have them in zoos, so you have to be careful about that. But they can scrape down to get to the grubs, and then they can pick them out with that specialized finger. So there's something else they can pick out with that specialized finger. Snot.
Oh.
Yeah. So they're breaking footage. Turns out the eye is only the 12th primate ever recorded to pick their nose and then eat it. Yeah. This is a study out of Switzerland's University of Berlin.
Oh, no.
And in the video, quote, the eyes inserts the entire length of its extra long, skinny, and highly mobile middle finger into the nasal passages and then licks the nasal mucus collected. Now, here's the thing about these long fingers. They're so long, they're like...
Yeah, that sounds dangerous.
It goes very far up their nose. So they actually used a CT scan of an eye's skull to try to reconstruct where the finger was going.
What?
And it turns out It's going down their throat.
Yeah.
One of the lead researchers says, quote, "There is no other possibility. Otherwise, it would have gone into the brain, and then they die." So, definitely going down into the throat.
They're not giving themselves a frontal lobotomy. They
No.
are scooping mucus out of, or they're itching their throat. I mean, what's happening down there?
Great question. The long story short is, we don't know. The scientists took this to go, why do animals pick their nose, and why do they eat it? And they wanted to do a research project where they looked through past research and they tried to compile enough information to formulate some ideas here. But they found that most existing literature was, quote, jokes.
Oh, geez.
So
Well,
this is an area where there's not a lot of research about it. And that at first they figured, oh, that's because it's gross, right? But how much research is there on coprophagia? Eating poop. A lot. So why is this a weird blind spot in biology? Great question. There's one study that suggested that nose picking could spread bacteria in a harmful way. Another one said that it could actually stop bacteria from sticking to your teeth by eating mucus, so it might be good for oral health. I personally feel like it's like itchy, it's like in the way, it's like flipping back and forth, like when you get your nose whistle, right? They get it out of there and then they're like, I can't, it won't come off. I know, it's gone. It's, you know, that's my guess,
maybe.
is that it's just their way of getting rid of it. It's easy solution.
Actually, you know, I think the reason there's not any research on this is because we know all humans pick their noses and eat their boogers.
What?
All humans, we all do it.
Justin, do you do that? I think you're admitting on air right now that you eat your own boogers. I do not do that.
At some point in time, children do, right?
Exactly.
All kids do.
Not in my memory did I ever do it. I thought it was so gross.
We'll get your parents on the show. We'll talk to them. So as a
I'm pretty sure they'll
father-
tell you the same thing. In fact,
as
they're
a
probably going
father
to
of
text me right
four-
now.
Oh, my poor kid. As a father of four, I'll tell you, some point, there's like a phase that can be anywhere from like a few months to several years. Children will pick their noses and eat their bogies.
Justin,
It's gonna
your
happen.
sample size has a lot of uncontrolled variables. They're
He has
all
a
yours.
Well, yeah.
He has more of a sample size than yours at the moment, Blair.
Except for the thousands of children that I've watched over the years in my career.
That's true.
Yeah.
Older children.
And none of them? So
No.
I guess
Some
it's
of them did, and some of them didn't.
Yeah. Or some of them got told not to do it in public.
You had
Like,
the
actually,
individuals who
literally
thought it was gross, and you had the individuals who could not get enough of it.
I can't believe we are debating
And
whether
hang
or
on,
not
and hang on.
the majority of people
And
at some point in their
the
lives have eaten
way-
boogies.
The way that the mammalian, I'll even almost say, nose is connected to the throat and mouth,
Mhm.
we all end up, occasionally coughing something, snorting. Yeah, there it is. It just went behind. If it goes behind, you know
Yeah.
that happens.
Yeah, that's fine.
That's fine.
Yeah.
If it goes the other, like it's so.
It's not intentional then.
Could be intentional.
No. But I get that.
Could
I
be.
get that. Like I would not the eating part.
Oh, I
If
got a
like-
response
if
from
I
my
had-
mother. She says I never ate my boogers.
Your mother is your biggest fan. I love her so much. I mean, aside from your dad.
I'm sure they're
Oh.
both screaming at the TV right now. Anyway, the whole point of this study, though, that was supposed to be a quick study, is that this is a blind spot in science, and this needs to be rectified immediately. We need to figure out why animals eat their own boogers, and definitely there are more that do it besides just these 12 primates that have been shown. So,
Okay, science, get to it.
get to
Study nose picking and boogie eating. Aye.
it.
says science, hopefully.
Was
But other news on nose picking in humans is that, hey, apparently, it might not be that good for you, just picking your nose generally. Researchers at Griffith University have published in Scientific Reports a study showing that chlamydia pneumoniae uses the nerve that's between the nasal cavity and the brain as a central path for invading your central nervous system. And then they showed, and you get the chlamydia pneumoniae could be on your finger because your finger's been all over the place touching things, and then you go and you stick it up your nose. And what they saw in their study that it sets off pathologies that are amyloid plaques. So beta amyloid protein becomes deposited as a result of the chlamydia pneumoniae infection, which is a marker that has been used for Alzheimer's disease evidence, although that is potentially something that is
very much
being looked at again. But anyway, so the question is, but the question is, if you're sticking your dirty fingers, if you're going to pick your nose, wash your hands first.
Yes, that's what I was going to say. So you got to wash it before and after then.
And after.
I have some important digging to do. I'm going to go give my 20 seconds, sing happy birthday twice, get that digging done.
Yeah.
Don't eat it and then wash again.
That's important to know because it's one of those things that feels like inherently dirty, so you might think, oh, I'll wash my hands after. But you're right. Because ultimately what you're doing is bypassing one of the best shields your body has towards foreign invaders, which are all your little nose hairs and your sinus. That's all there to protect you from germs. So you're bypassing that by sticking your nose up there.
I guess that's why you've got all the mucousy stuff in there, is to catch things before they hit the membrane that you also use to smell with and stuff.
That's exactly it. Because your olfactory nerve is directly connected straight into the olfactory bulb in your brain. And so if you get something infecting those nerves, that can actually get past the blood-brain barrier and into your brain and cause disease. However, this study was just going to say, though, this study so far, it's a dish. Neurons in a dish. We're also talking about a mouse model. We're not talking about actually having seen this in humans who have picked their nose. There is no absolute link between picking your nose and the pathogen and developing Alzheimer's and or dementia. This is all still speculative and in just initial research phase. So I just want to put that out there. But it is something to think about if you're putting a dirty finger up your nose. What else is going up there?
It's not just what's coming out. The human trials are going to be impossible because there's no control. You can't find a human who's never picked their nose.
Let's get your mother back on here. I think we found one.
I never said I didn't pick. I just said I didn't eat.
She is a test to the fact that my daughter has never picked her nose.
Never.
No, man. That's going to be so hard, though, because you're also going to have to ask people, even if you don't have a real control, you're going to have to ask, how frequently do you pick your nose? Nobody's going to be able to answer that correctly.
Forever, it is going to be a correlation, and I mean the absolute causal relation in humanity. I know. Is your nose itching? Mine's a little sniffly at the moment. Justin, do you have another story?
Oh, yes. Okay, so I want to do the DNA of the ancient South American story, but I'm going to do it and why Americans die so young. But I think I'm going to jump to, so we can get on with the show, deeper into the show. This is the COP27 climate change conference is coming up.
It's going to
be happening over the next week or two in Egypt. Here's hoping that something real, meaningful, consequential comes out of it. Really, I hope so.
Fingers crossed.
Researchers at University of Melbourne examined official climate plans and past public statements, including some of the nationally determined contributions which countries submitted to the United Nations as part of the Paris Agreement, to calculate the total amount of carbon offset that they would be doing. They listed one of the things is we're going to dedicate land to planting trees. That's a good thing.
Yes.
So basically, a plan to grow more trees as a carbon offset. Fantastic plan. But these folks, this group at Melbourne, I think the University of Copenhagen was involved in this as well as a co-author. They added up the amount of land proposed to be used for growing trees. And calculated that countries collectively needed a total of about 1.2 billion hectares of land to fulfill the promises laid out in the official climate plans. So
that's a lot of space.
1.2 billion hectares, it's about 3 billion acres of trees that they would be planting to get to the carbon offsets. Now, it's a big number, and it's difficult kind of units to conceive of. So let's put it in Rhode Island, which is the preferred measurement standard used to describe like big glaciers, the occasional massive asteroid out there. Oh, it's this many Rhode Islands, right? Let's see, that would be 3.8 million Rhode Islands.
Ha!
Okay. The United States, including Puerto Rico, is about 2.3 billion acres, which is less than the 3 billion acres. All of the United States. And now, if you know, the United States, also, you can't grow things everywhere. All of Europe is about 2.6 billion acres. All of Europe combined. Still less than the 3 billion. And not all of it is, you know, there's places where there's trees already. There's places that are deserts. There's places where there are cities. There are places where there are farms. There are a lot of the land is actually used for things. So
what do we do? I mean, how do we do this? It's a lot of trees.
So spread out around the world, and assuming that you don't cut down trees to plant the carbon offset trees, and you don't get rid of the cities or plant in the deserts or on mountaintops or places where trees don't grow, it would require half of the current global croplands be converted to forest.
So now we pick who's going to starve first? Like
kind of. And most likely it's going to be people who have probably an indigenous claim but lack political leverage over their lands. Somehow, in order to make the numbers work for the showing carbon offsets, the nations have been using this massive scale industrial planet-wide tree planting accounting program to show how they are going to get to the carbon neutral point. So let's get started. Where are these super nurseries? Where is the call to mobilize the youth of the world to enlist in the arborist forces? Where do they report to? Root camp. Okay, enough. The report cites a growing body of evidence showing that when indigenous peoples and local communities have secure land rights, they vastly outperform both governments and private landholders in preventing deforestation, conserving biodiversity, and producing food sustainability. Conversely, the report also alludes to the fact that, as I was saying, many indigenous lands could be impacted if nations actually went through with their phantom accounting, with this global mobilization towards tree planting. But the point is, they haven't started, and they're going to plant a forest larger than all of the United States.
Like in the next, 20 years?
So what you got to do, here's what you got to do. I figured it out. I don't know why no one else has thought of this. Okay, so you plant trees on top of buildings.
That's still, actually, if you just took the buildings out of it, it doesn't change the amount of land that much. It's still It's more
We could plant trees on the sides of buildings.
planting
too.
trees on
Ooh.
tops.
There you go.
Vertical
Vertical
forest.
space. Yes.
Plant trees on top of the existing trees is what they would really need to do. Get
That
really
was
good at
that was recommended in our YouTube chat by Jason Wagner, saying we need to think like an engineer. Nothing says you can't grow a tree on top of another tree.
Oh yeah, there it is, Jason. Good job. So
There we go.
Yeah, so basically
Trees everywhere, and you get a tree.
We've been getting, like, at some point somebody figured out, it's like, actually, if you put all of your carbon into trees you're going to grow someday on land you don't have, it makes it look like we're complying. Oh, I'll just do
It's
a
greenwashing.
ton of that.
Yeah, it's a lot of greenwashing, and that's, we've talked about all of this, these, the glad handing and the talking and the promises being made, where, all sorts of manufacturing companies the top beverage companies promised to use less first use non-recyclable plastics in all of their products. And the amount of non-recyclable plastic is actually increasing in their across the board in their products. So, I mean, this is, they're saying one thing to you while they're doing another, and they're playing a shell game that is going to wind up, it'll make people's pocketbooks really big for a little while, and then we're all going to suffer. So
good job with the clear-eyed folks at Melbourne University and University of Copenhagen for getting together and adding up, the promises.
Add it up.
Yeah. Hey, so maybe just a quick thought I'll plug to kind of button this. There's a pretty big election next week in the United States, and I'm not telling anybody how to vote, but, if this is something that you care about, look into who's actually interested in doing real things to help with climate change and support those people.
Just like they just recently did in Brazil.
Yes.
Yeah.
Yes. So if everything goes well, the incumbent has promised to decrease deforestation in the Amazon, which is huge. That's the kind of action.
It was actually increasing before.
Yes.
They cleared all that land so they could plant trees for carbon offsets. It's all part of the plan.
That makes no sense, but or it's either that or palm plantations. But anyway,
it's another side point, which is that if you left a forest undisturbed, it does better as a carbon sink than if you planted a new forest. Anyway.
Yeah, of course. Yeah.
Math,
Yeah.
Oh, side points and side things and big corporations and other things the big corporations are doing. Meta, the company that used to be Facebook that is trying to create the metaverse, we know that they're not just working in VR, they're also working in AI. I love all these algorithms. Recent article preprint out about their AI effort. They have taken a hint from the London-based old Google-supported, Alphabet-supported DeepMind research, AlphaFold, that has been involved in predicting protein structures and nailing down protein sequences in the known gene and protein databanks. Well, they have just described, the Meta AI team has just described in a preprint using a large language model, which is an AI that uses that has a basis for tools that can predict text from a few letters or words. So they're using language models to look at the sequences of amino acids and potential mutations and different changes within them, and trained them on the unknown dark matter of the microbiome. And so there's this Meta AI called ESM Fold, which isn't quite as accurate as the AlphaFold AI. It's really, really good, though, at predicting structures and for kind of guessing what mutations might be taking place. And so it predicted the structures of more than 617 million proteins in metagenomic DNA from environmental sources, including soil, seawater, the human gut, skin, and other microbial habitats, describing proteins that have never been sequenced before, describing proteins and predicting the structure of proteins that have never been described or predicted to exist before. So it is a really interesting step into that undescribed dark matter of the world of DNA that surrounds us, that influences us, that has impacts like Justin, you were talking about with the, microbiome of the gut of the fish developing social behavior in the zebrafish larva. This effort took 2 weeks. So 617 million predictions.
Wow.
Yeah. Anyway, it's a very interesting effort, and it'll be interesting to see where these new models of AI take us in our understanding of the world.
And how will they be turned into profit?
That's what I'm wondering as well. Like, what are we going to do with this? I mean, medicine and other things, that'll be great. But, hope maybe in the end, what it'll help us do is reconstruct our biomes because we've destroyed them all. So anyway, time to move on to more happy topics.
Mm-hmm.
Well, that is a very interesting point, though, because there has to be, when we talk about IP, when we talk about patent walls around things, you have AI that came out and how many thousands, tens, thousands of things that it named. It's like, I named all of those and I discovered them and therefore I own them. Like, but what do? That's not later, maybe. Like, that can't be how we move forward with anything.
Yeah.
Yeah.
Well,
so,
it's the production. It's the actual production of them, and meanwhile, we still have to actually confirm a lot of these predictions that have been made for the structures. Understand the functions. Possibly, there's a lot of confirmation and refining that needs to go on in the future.
Yeah, of course. But there's like that classic case. There was somebody attempted to patent a gene involved in breast cancer.
Only if you can make it, right? Isn't it? It's like if you are the you can patent the manufacturing process of something, but biological components that are in existence in nature, you cannot patent.
Yeah, but if you make an alteration, you can patent to an extent. So it's like it gets very
synthetic DNA.
Mm-hmm. And then, I just put an extra nucleotide, so now it's a completely new product? Does that even make sense? Like, at some point,
I will own this whole country because I will have made my genetic mutation possible. It's going to have gene drive to take over the entire population to make the population less sick and live longer, but you all are mine.
You'll have to pay a monthly subscription fee now.
$8.
Yeah, $8. But then in a few years, I'll change it to $12.99.
But then a lot of people will drop out,
and then we might.
Like, oh no!
We got to put ads. We're gonna have to put ads in your DNA too.
Yeah, for sure.
A free ad version of your own DNA. Oh gosh.
This is This Week in Science. Speaking of ads, this is not an ad. This is just a reminder that you are taking part in this scientific discussion with This Week in Science. If you're enjoying the show, please share it with a friend today. Time now for that wonderful time in the show where we get to learn about animals from Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's animal for
what you got, Blair?
I'm very excited to tell you about Playful Bees, but I'm saving it.
Ooh.
First, I want to tell you about some smart jays. So jays, as we know, are in the corvid family. So they're very, very, very smart. They're related to crows and ravens, and they're like toddler level smart. So they're really interesting to study. And so this is a University of Cambridge study looking at Eurasian jays and a version of the marshmallow test. So I thought it would be fun to talk about that because this is a recurring test that we just keep bringing up on the show. All of the loyal listeners know all about it, but just in case you haven't heard of it, the marshmallow test was developed in 1972 by Stanford, and toddlers were offered a choice between 1 marshmallow immediately or 2 if they waited. And essentially, they were testing if the children could delay gratification for a greater payoff.
They thought they were testing for that.
And generally speaking, see, this is where it gets complicated. Generally speaking, when they followed up those tests with general intelligence tests, they jived. So if the children did well on academic tasks, they were also the ones who were able to resist temptation for longer. We know that these tests are somehow problematic now for a bunch of reasons, mainly because you can't test humans like you test jays in a laboratory, and there are lots of other confounding variables at play. They found out...
Yeah, okay.
Go ahead.
Well, because every time I hear about this test, I start chomping at the bit, be like, that's not what they tested! That's not what they were testing. They thought they were testing that, but what they were actually testing is the socioeconomic background of these, or environment of these children.
That was part of what they were testing.
They weren't.
But regardless, they were testing self-control.
No,
they were testing self. You're right, but they were testing self-control.
Positive though. They thought they were testing self-control. Hang on, just give me a second. They thought they were testing self-control, but they were accidentally testing the socioeconomic strategies. If you're a kid who was in a poor environment, the best strategy to employ is to grab that treat when you can, because there's not more in the cupboard. If you come from a household where if you pass on the treat now, but you can have it in an hour or 2 hours, it's always there. Then the strategy isn't delay. The strategy is, I trust that it will be there. My strategy can be get more. So it came down to...
So, Justin, the problem with that...
Those kids were, the impulsive kids were actually using the best strategy for them based on their experiences. That's the whole... When they redid this test years later, that's what they discovered they were accidentally peering into. But it's not that they were testing that. It's that was a confounding variable. Oh. So basically, so they're still ultimately telling these children, if you wait, you get 2 marshmallows. That does not change. And regardless of what your socioeconomic status is, it is the better choice to wait for 2 marshmallows. The problem is that if they were raised in a lower socioeconomic status, they had been conditioned to act differently.
Which is the confounding variable.
That is a confounding variable. That is not a test situation. So that is the difference here. They are still testing delayed gratification. They are still testing this situation. But because of socioeconomic that's just how variables work. Because of the socioeconomic status, that is a confounding variable that had conditioned them previously. They had previously been trained to act a certain way in relation to food. That means that there was not a clean slate for the children entering that room. That is the issue, because all of those children were told the same thing. If you wait, you get 2 marshmallows.
Okay, but that's the marshmallow test, and that's in the human version. But Blair, you want to talk about jays.
Yes.
Which were probably just in a laboratory for like their whole lives.
Yes. And jays or other corvids are an excellent choice for this study because they already cache things. They hide food, they save it for later. So they are already well-versed in delayed gratification. They won't eat it now, knowing it'll be there for a future meal. So the researchers thought that this might have driven the evolution of self-control in these birds. And so they wanted to study their ability to do this kind of self-control test. So what they did is they presented them with mealworms, bread, and cheese. Mealworms are their favorite. But bread and cheese come in second or third, but that varies from individual to individual. Everybody loved mealworms. Bread and cheese, everybody had a different second. Right. So they had to choose between bread or cheese available immediately or a mealworm that they could see, but they would be given access to after delay if they did not eat the bread or cheese. So the range of delay times was tested all the way from 5 seconds to 5.5 minutes. There were some that never got past 20 seconds. J-Lo, the top of the class, went to 5.5 minutes waiting for a mealworm. But the bottom line is all of the birds passed. They all of the jays in this study were able to delay gratification for their worm. They also looked away from the bread or cheese when it was presented to them. Also, almost like they were trying to not get distracted or tempted by the food, which is exactly what chimps and children do in the study as well.
Yeah, they do that too. They try not to look at it.
Yeah.
They're waiting.
I'm not gonna look at it.
Yeah, and then the other thing that they did is they adjusted self-control according to circumstances. So if they took the worm and it was out of reach, so they knew there was no way they were ever going to get it, the jays always ate the bread or cheese if it was available. So they could recognize, I'm never going to get that. I should eat this food now. And the length of time they were willing to wait fell if it was pitted against their second most preferred food. So if I'm J-Lo and I go, mealworms, cheese, bread. If you give me bread, I'll wait much longer for that mealworm than if you give me cheese. It's more tempting.
Preferences matter. Yeah.
So that shows that jays only delay gratification when it's warranted, so they can kind of weigh these different options. And then the important part of this study was looking at cognitive abilities of these birds, because they wanted to see if intelligence was related to the ability to delay gratification. And so they gave the jays 5 cognitive tasks commonly used to measure general intelligence, and the birds that performed better in those tasks also managed to wait longer for the mealworm reward. So that suggests that self-control is in fact linked with intelligence.
Never mind. It is a very limited number of birds in the study.
It's a limited number of birds. But it's one species, and as I mentioned, it's a species that already caches their food.
Yes.
So they're predisposed to do these sorts of things. Yes.
So
absolutely.
here's what you got to do to prep some of these birds for the human test. You got to put them in there with 2 other hungry birds and put the 1 piece of cheese. And now they can wait for the mealworm, but if one of the other birds eats the cheese, they get nothing. That's
a bird massacre is what you're going for?
No, that's what the human socioeconomic background strategy for getting the thing as soon as you can is if it might not be there later. If
right. Well, they didn't do that with the children in the labs either, but that's okay.
No, they did. They did actually. It was already taking place in those children's lives. That's the point I'm trying to make.
That's okay. We're back to the difference between an experiment and a confounding variable. Anyway, moving on to
a conclusion.
Bumblebees who play. Yes, this is a study from Queen Mary University of London. And they found that bumblebees love to play. It's the first time that object play behavior has been shown in an insect, and so they indicate that this may mean that bees experience positive feelings, which, no surprise, I will just say, duh. Anyway, bees went out of their way to roll wooden balls repeatedly, despite there being no incentive to do so. Younger bees rolled balls more than older bees. This mirrors human behavior of young children and other juvenile mammals and birds being more playful than adults. And so what they did is they followed 45 bumblebees in an arena. They gave them options of walking through an unobstructed path to reach a feeding area. So just here, straight shot to food. There you go. Or deviating from that path into areas with wooden balls. So going completely out of the way of where their food is, to go play with wooden balls. Individual bees rolled balls between 1 and 117 times over the experiment. So
there's the little sports bee player there.
Exactly. Yes. Well, it's interesting you should say that, because in a previous study, bumblebees were taught to score a goal.
Oh.
So they were taught to roll a ball to a target in exchange for a sugary food reward. But so they showed that the bees could do this and could figure out that it was something that they could do. But where this becomes special is that these bees were not taught to do this. They were not conditioned to do this. They were not given a reward for doing it. There was no benefit to them playing with these wooden balls. It's voluntary, it's spontaneous, and as far as they can tell, it does not mimic any behavior that they do in the wild that they would be kind of like practicing here. In a further experiment, they gave another 42 bees access to 2 colored chambers, one always containing movable balls and one without any objects. When they were given a choice between 2 chambers and when they were both empty, bees showed a preference for the color of the chamber that previously had all the wooden balls in it. So they were like, where's the ball pit? I want to go play in the ball pit.
That's fascinating.
Yeah. So despite what people often see as teeny tiny brains, barely even brains, nerve balls, they're more than just kind of simple command and response animals. They appear to have emotional states. There's something that they find rewarding about playing with these little wooden beads. And so this does potentially have impacts on our understanding of sentience and welfare of insects and emotional status of insects and other invertebrates and other, quote unquote, lower life forms, right? That we don't kind of associate these kinds of emotions with. So there are lots of animals who play just for the purpose of enjoyment, but most, of course, have been recorded in mammals and birds. So this is something that is really unusual to see the bee doing that. And if you are listening to this, please go to our show notes and watch these videos because I want this on loop just around me all the time. It's so good. They're just having so much fun and they're bumblebees,
really
so they're
do
fluffy.
Look like they're having a blast.
Okay, so I'm gonna question the idea the idea that they're playing. I mean, is it play or is it just interaction with a more complex environment that is somehow interesting to them? Does it have to be quantified as play?
Right. So I guess that's the question, right? Is there any other motivation that could cause this? Is this some sort of exploratory behavior that can give them an advantage? So I think you're right. There needs to be a much more robust study of bumblebees in the wild to see if there's anything at all similar to exploratory behavior, tactile behavior like this, to see if there's something related.
But I mean, we haven't been invited to the bee soccer matches before,
No, I don't think they go pro either. So, but my question is, isn't that kind of a good definition of play? That sort of tactile exploration?
Yeah.
To, on some degree, I mean, I think that would be a sufficient enough to call it play. I wouldn't call it an organized sport that they're involved in.
Right. Well, and then, that's a really good point, because even if it provides an evolutionary advantage, you could argue that definitely human play when you're a child provides an evolutionary advantage because you are learning socio-emotional cues. You are learning in lots of other ways. You're exploring your environment. You're building social interactions. So there's evolutionary advantage to a lot of types of play. You're getting smarter by playing with blocks when you're a kid, right? So there's all these things that you're doing. You're learning to count. It's lots of things that come from play that are beneficial. So you're right. It gets muddy. And this is probably one of those things where scientists love to put terms on things and we want to call this play, but what does that really mean? Does it just mean unproductive behavior? Yeah. Well, it's not productive for any purpose that we understand or that we comprehend. It's not something that was necessarily conditioned or learned. It wasn't something that the researchers went, you must do this for a reward. And that it's fascinating that the bees were choosing the balls, and not just the chamber with the balls, but actually to interact with the balls. Yeah, I think there are a lot of interesting questions to be investigated here because, like you said, there's so much that it could
lead to in terms of understanding insects and a lot more about the world that we live in.
Right. I think about when I was working at the zoo, we'd have the feeder crickets, and we'd put egg cartons and toilet paper tubes and food that we knew they wouldn't eat in their scraps of other food, right? For them to crawl all over and be on, and we changed that over time. Why did we do that? That was enrichment. Why do crickets need enrichment? It's animals need novel things. They don't like to be bored.
That's what this show is all about.
Enrichment for everyone.
Yeah.
It's one novel thing after another.
Yeah.
Be footballers. Who knew?
There you go.
Maybe they're just getting ready to roll their little bee larvae around.
Mm-hmm. I like to think it's bee croquet.
Those are pretty big, though. I mean, it's almost like when we were watching those videos there, the bee is almost the same size as the ball. And sometimes they're kind of getting tumbled over with it when they're playing with it, and they're right back to it. So it's not also like a super casual activity.
No,
they're completely involved, expending energy doing this. Yep. That's, I think, to me, why it looks
like a log rolling championship at the Lumberjack Olympics.
Yeah.
Which, if that were to be studied by alien scientists, you'd be like, why are they expending energy doing this? Yes.
Could they be playing? No, that's not possible. They're humans. They're big hairless apes. But so there's no analog that I can think of too in nature where they would be specifically doing this large object rolling thing. But that's also what would make it novel to them, I suppose. That they haven't encountered.
Now I want to know about spider play. I know spiders are playing.
Oh, they are.
I want to know.
That's what they're doing when they're building a web right in front of my face. They're playing with me.
They're playing with you.
They're doing trick or treat.
They're snarky. Bunch of snarky spiders.
Yeah. So snarky.
Oh,
this
did I make
This Week in
web
Science.
at face height just outside of your back door? Oh, is that me? Oh.
This is This Week in Science. Thank you so much for joining us for another fun-filled episode. We've got, we'll talk about spiders and bees and all sorts of things. And we hope that you, if you appreciate it, head over to TWIS.org and click on our Patreon link, because that is how we are supported, is through our listener support of our patrons. Choose your level of support, $10 and more per month, and we will thank you by name at the end of the show. That's TWIS.org. Click on that Patreon link and pledge your support to TWIS. We can't do it without you. Thank you for your support. All right, Justin, back with more This Week in Science. What do you have to tell us about?
I've got DNA from some ancient humans discovered at different archaeological sites in South America, down in Brazil, Uruguay, and Panama. So this is Florida Atlantic University and Emory University researchers. They use DNA from some current and past studies. Findings show a distinct relationship among ancient genomes from northeast Brazil, southeast Brazil, Uruguay, and Panama. The model suggests, according to the researchers, the settlement of the Atlantic coast occurred only after the Pacific coast side had been settled, and along a south to north migration route. The ancient individuals from southeast Brazil are about 9,000 years older than those from northeast Brazil, Uruguay, and Panama. Thereby, that's their illustration of migration direction. They discovered evidence of Neanderthal ancestry in the individuals. Very interesting. Researchers also detected greater Denisovan than Neanderthal ancestry in ancient Uruguay and Panama individuals. Which is, I don't know if you've got a map in your head of South America. Uruguay is kind of dead centered and a little bit to the south-ish. And Panama is Central America, basically. You have to go all the way up through Brazil, you got Colombia there, you got to go up before you even get anywhere near Panama. So these are two closely related. They said significantly related, but geographically well dispersed. They say the admixture, this is according to John Lindo, assistant professor of the Department of Anthropology at Emory University, one of the co-authors of the article, who specializes in ancient DNA analysis, says the admixture must have occurred a long time before, perhaps 40,000 years ago. The fact that the Denisovan lineage persisted and its genetic signal made it into an ancient individual from Uruguay that is only 1,500 years old suggests that it was a large admixture event between a population of humans and Denisovans. And they got a strong Australasian signal in an ancient genome from Panama, which could be really significant because it hasn't been seen at all in North America. I guess it wasn't seen, that strong signal wasn't seen in the 10,000-ish year old sample to the south. Actually, so this is, but this whole thing has got me wondering if that could be a misleading hit. So if you took DNA samples from a remote coastal Norwegian village, one that had been pretty well isolated for like a really long time, like nobody goes up to that town, nobody comes from that town, they're up there, they fish, they go home, that's kind of it. You might be surprised to find out that the residents there are 15% British.
Yeah,
right?
A little surprised.
Only history likely then informs us that the genes they have identified as British more likely have come from Viking raids 1,000 years ago and dispersed into what is now the UK. Much more likely than it is from an unknown populating of rural Norway by seafaring Brits. Right? Yeah. So I'm thinking that it's possible. I think this study needs to get interrogated a little bit. I'm thinking the Australasian hit could actually just be Denisovan, because that's also where the signal of Denisovan is the strongest in any existing population, is in that Oceania area. So something to me sounds sort of like the Norwegian British by getting all the way down into the Americas and having this hit of Australasian. Because the other scenario is that an Australasian cross-Pacific settlement event occurred somewhere near Panama over 1,000 years ago, which could actually account for the higher Denisovan DNA in the current population if they were highly admixed before crossing. And that is where, so there is an argument that you could make for it as well. Although,
but then there's also
would be the more interesting.
Yeah, but then there's also the question of, is our analysis of these various admixtures, what is the accuracy of it, and how much are we really seeing the mutational and crossbreeding
right.
paths versus times in our history in which peoples were wiped out, or there,
that's a great point, because it's what is it? Is it the Upper Dryas? Is that the one? Hey, there's a major bottleneck event that happened. I think it was 1,500 years ago. We talked about this on a story, some months ago, I think. So there is a significant spike of identifiable genes in certain populations after that point that would have been, not as signature before. So you can sort of picture a group that has a little bit higher Denisovan than the rest during a bottleneck event isolating as they traveled to a more rural part of South America, surviving this event. Maybe those genes help them survive. Who knows? But now you have this higher signal population that itself gets dispersed after that. But so there's a lot of ways that this needs to get interrogated before you start connect. Because gosh, every time we've tried to connect the dots on limited information about the Americas, we've been wrong. Science has gotten a lot, getting there.
Yeah, we're getting there.
But there's been so much bad. But here's and this is Lindo points out. It was the researcher from what was it? Emory University. Lindo points out that only a dozen or so ancient whole genomes from South America have been published to date. So in Europe, there's many hundreds, and it doesn't even go back as far, right? Like the current modern populations of Europe, maybe 5,000 years. So this is going back to 10,000 years for the one individual. We know there was people in South America long before that. But we don't have a whole lot. We have a very small sample size to work with as well. So even the migration route, even all of it needs more digging, as is always the answer in archaeology and anthropology. More digging is required. Get this. Results of the study is published in the Journal Proceedings of the Royal Society B for anybody interested in looking at the complete study. And then my final story for the night is Americans die younger than people in high-income countries.
Why?
Well, there's an average life expectancy of 78.8 in 2019. So this is before the pandemic.
Recent events, yes.
Before the recent events. Which means Americans die younger than people in Japan, but everybody dies younger than the people of Japan. Americans die younger than the people of Canada because of their fitness and food selection. Also, the UK. In fact, actually, you have to go all the way down the list of nations' life expectancy. We fall between Cuba and Albania, the United States does, which are considered middle-income countries. Now, that's if you look at the USA as a whole. If you look at it by state, there's a strange disparity, a range of ages, depending on what state you happen to live in. The high end is Hawaii. 81 years. In Hawaii, so you get a little over 2 extra years. Almost 3 years. 2 extra years. If you're living, than the average, if you live in Hawaii. If you happen to live your life in Mississippi, it drops 4.5 years, down to 74.4.
4.5 is a lot.
It is a lot for averages like these, yeah.
Off of the average, which is also already below the rest of below all the high-income countries. So evidence from previous studies consistently pointed to heart disease, drug poisoning, suicide, alcohol-induced deaths as key drivers of high mortality rates among working-age Americans. According to this study published in PLOS One, Syracuse University finds that more conservative state policies were generally associated with higher mortality. Researchers used data from 1999 to 2019, again before the pandemic, National Vital Statistics System to calculate state-level age-adjusted mortality rates for deaths from all causes, and specifically those of cardiovascular disease, alcohol, suicide, drug poisoning, amongst adults ages 25 to 64. They merged that data with annual state-level data on 8 different policy domains, where each state's policies were scored a binary 0 or 1, whether it was conservative or liberal. The analysis revealed that more liberal-leaning policies on the environment, gun safety, labor, economy, tobacco which is the taxes on tobacco and where you could smoke, I guess, were associated with lower mortality in each of those states. And a particularly strong association was found between gun safety policies and suicide mortality amongst men. Between labor policies and alcohol-induced mortality.
Labor as in work?
Work.
Yeah.
So
okay.
Work policies, safety, hours, wages, that sort of thing, and alcohol-induced mortality were linked. Links between economic and tobacco policies, tax policies. So tobacco is more expensive, I guess, in the liberal-leaning states because they tax it higher. There's a link between those policies and cardiovascular disease mortality. So they had all this nice data, and they decided to run a simulation. Simulation suggested that changing all policies in all states to a fully liberal orientation of policies could have saved 171,030 lives in 2019.
Just in 1 year.
1 year. Changing them to a fully conservative orientation would have cost an additional 217,635 lives. All this is before COVID. All this is before. That's a huge disparity of 400,000, nearly 500,000. Right? This is without COVID in it. So anyway.
But in a way, COVID was kind of a test of this in a really, really bleak, terrible way, because we had federal level decisions being made that were highly conservative. So we lost lives.
Different states implementing public health measures based on their local practices and their local regulations. And so that we saw a difference in life expectancy in the last couple of years between states as well. So
just quick to point out, education levels, income levels, healthcare access were not specifically looked at in this study. Though if I were a guessing man,
I would do that.
I would probably guess that educated people with resources, access to doctors, probably live longer than those without. Could be wrong. Follow-up could find that poverty, educational exclusion, and a lack of healthcare make you live longer.
I'm gonna guess not.
See, all these things are linked too, right? Though, so these are directly along the same lines. You have a lot more access to higher education in bluer states. You have more access to healthcare in bluer states. So there are more economic safety nets in bluer states. So you'd have a lot of those same things kind of leaning in that direction anyway. But I do think, as Kevin Reardon brought up in the chat in a few different ways, the thing that makes us so different from other quote unquote wealthy countries is that we have a pay to play insurance structure. And so all of these things aside, if you're sick, you have to decide whether you can afford to go to the doctor. And it is not like that in any of the other countries that you mentioned, Justin.
Our neighbor to the north, Canada.
Yeah.
Front BB wants to know, where should I move to? Hawaii.
Hawaii.
I mean, that's the right answer for a lot of reasons. But if you're thinking of this study alone says move to Hawaii.
Hawaiians might tell you don't move there because they're full.
Probably. It's like New Zealand, right? They're like, we're done now. Okay, we got enough of you. You could go now. I have a couple of stories. Were you done with that one, Justin?
Oh, yeah.
Did we play it out?
What do you have, Kiki? You have some gross stuff, don't you?
Oh, yeah. I mean, we're talking about all these politics, and I know sometimes people hear about politics on a science podcast, even though we're discussing scientific analysis and science policy and public health and that aspect. Still, I know it makes some of you retch and maybe want to vomit. Maybe your stomach's just wanting to reject all of this. What makes that happen? What is it? What signal from your gut, from your brain, makes your body decide it is absolutely going to reject whatever is in the gut cavity.
I know.
Yes, Claire.
Yeah,
Claire.
Osmosis Jones hits the vomit button, right? Do you remember that movie?
Yeah.
What are you talking about? How come I
Throwing
feel
it
like
back.
we've lived in completely different media environments?
Sorry, continue.
What? Wait, no, let's stop the show. I need to find out who's Osmosis Jones.
We'll talk about it later.
Oh. Yeah, you do need to find out who Osmosis Jones is, but for right now, we're going to be talking about really what happens in the gut in the gut brain axis when it is time to vomit. Historically, we love looking at mice because mice are little. We can have a lot of them in a laboratory, and they're great to be able to look at their brains. And there's so many pathways that are very similar. And it's like, yes, it's a mouse model of mice don't vomit.
Mm-hmm.
They can't vomit.
They can't?
There's a bunch of animals that don't really do that.
A lot of animals cannot.
Yeah.
So dogs can, cats can, but you don't put dogs and cats into laboratory research in the same numbers, in the same way. And the restrictions around research related to dogs and cats is a little bit different to those around experiments related to mice. And so we haven't really done the work to dig into the gut-brain axis related to vomit. Emesis. How does it happen? Well, these researchers publishing in Cell this week their paper, The Gut-to-Brain Axis for Toxin-Induced Defensive Responses, they have determined that they found a way to model vomiting in mice. So they discovered that if you give mice a toxin that is produced by Staphylococcus enterotoxin, which is called staphylococcal enterotoxin A. If you give them that toxin, then mice open their mouth a little wider than normal, and they make a little movement in their mouth that's as if they're retching, like they're dry heaving.
This
is
a much worse version of if you give a mouse a cookie, and I don't like it.
If you don't give a mouse a cookie, but instead give them an enterotoxin that's bad for their gut, they will dry heave.
All right, don't make that sound, Claire. This is a very...
Okay, so in this, what they discovered is the enterotoxin leads to the release. It interacts with the lining of the gut and a particular type of cell in the gut called enterochromaffin cells in the lining of the intestinal lumen. These cells then release serotonin. Serotonin triggers the vagal nerves, very specific neuron leading into the dorsal vagal complex in the brain. TAC1 DVC neurons in the brainstem, they get triggered, and they discovered when they inactivated those neurons in the mice could no longer retch. And then they were like, so what does this mean for chemotherapy drugs that we might give people to control their nausea? When chemotherapy makes people nauseous and we give them a drug that reduces their nausea, what is it actually doing? And so they gave the mice a drug called granicitrin, which works by blocking serotonin receptors. And they found when in inactivating the TAC1 DVC neurons, it blocked the response. And so they were able to determine that this is exactly how this granicitrin works. It blocks serotonin receptors. It blocks the pathway. And so it blocks the your urge to purge.
What about for morning sickness? Would this help with that? Like severe case of morning sickness?
Right. So these anti-nausea medications can work for severe morning sickness, but they are not. This is where things have gone wrong in the past, because this is where the drug that was given many, many years ago that led to severe birth defects in a number of people in Europe.
You got to be careful
drug-
with those
and in the United
pregnancies.
States. And in the United States.
I thought it wasn't in the U.S. I thought it was just in Europe.
I know people.
Okay.
Who
the way
have been
the way that the drug, however, was an anti-nausea drug, and so side effects are very important. And so that if you're blocking serotonin receptors, what is going on? Are you blocking other things?
Right.
Yeah, Zofran. I've had Zofran before. Zofran, thank you, Rick Loveman. Zofran is one that's given very commonly to children to calm nausea, and when you're sick to your stomach and you have a stomach bug, like what's the one that goes around in the wintertime with the kids that we don't like very much anyway. If you're vomiting a lot, you can take the Zofran and it keeps you from vomiting too much, so that you can actually start to take food in, so you can get nutrients in and not just become extremely dehydrated and undernourished while you're that ill. So the big goal in this study, why do we want to make mice vomit? Why do we want to do this? We don't want to do this, but what it's telling us is potentially the pathway. So now we have a target in the dorsoventral or the dorsovagal nuclei in the brainstem. We've got a serotonin receptor. We've got other things that we probably need to be looking at those enterochromaffin cells to really understand more about how they're being triggered and what's triggering them, what specific toxins trigger them, how does that actually get the process started. And in the case of nausea and other issues that are not toxin related, where you maybe are nauseous, but we don't know why, these pathways need to be determined so that we can create more drugs to combat nausea, to combat the vomiting, to be able to allow people to, take drugs that are more necessary for their survival, like chemotherapy drugs. Yeah, and stay healthy. So anyway, vomiting. I'm glad we all made it through that particular
This might also be handy for med school.
If you need drugs to get through your anatomy class, you probably shouldn't be in med school. There are far worse things waiting for you on the other side.
I'm going to be an ear doctor. This is the last time I have to look inside a thing. Oh, it's the inside of the ear also? Oh, man.
Understand this pathway. So, while we're trying to map the internal spaces of the human body, man, there have been animals out there, even humans, who have been mentally mapping physical space using sound, sonar, right? And MIT just reinvented sonar, but with AI. And so they've developed a machine learning technique that accurately captures and models underlying acoustics of a scene, of a room, of an area, and can do this modeling from a limited number of sound recordings. So you can go into a space, make some recordings of that space, and then this machine learning technique is able to put it all together to computer model, and generate the spaces more accurately than has been done before. So the bottom line here is that these researchers at MIT and the MIT IBM Watson AI lab are probably going to be they're developing this 3D generation of the geometry of a room from these sound recordings, so that can be used in VR, right? Can be used in gaming, can be used potentially. I love the idea of having a concert hall being accurately acoustically generated and being able to sit in VR and listen to an amazing classical piano performance, or something like that, or your favorite rock band, or whatever it might be. This kind of research is going to lead to these possibilities. And so these researchers who have described the model, they talk about how a lot of modeling has relied on vision so far and not so much on the auditory environment. However, as humans, the audio environment is so important, and it's something in video production. It's like if your audio is bad, people aren't going to watch your video, right? So having really accurate audio can only make our imagined spaces, our modeled spaces, that much better.
So is this like, okay, we got a call from the what do you call it? The kidnappers. They want the ransom money. And then you play the recording through the AI and says, oh, this is in a 2-bedroom split level. The only way this recording could have been made is a 2-bedroom split level with an east-facing window that's got a sliding glass door. And then you narrow down how many of those are in town, and the good guys show up and they rescue me, who got kidnapped and trying to be ransomed. But because they're able to write the room,
yes,
describe the room based on this. That is super fascinating, and also a little scary. This is falling into that category of the lie detector that they can play over speech.
Mm-hmm. Exactly.
That's terrifying, because anybody who's been recorded talking, they can tell if you're lying through AI in the future. Now you're saying that they can also tell what room I'm in.
Right. So from the recording of a space, they can, they're beginning to, it's not just, so from PBC saying, right, there's a SIGGRAPH paper from decades ago, which was modeling the acoustics of a space. We've been doing this for a very long time. Yeah, however, this is getting to the point where if you take a couple of recordings from a space, you'll suddenly be able to acoustically know where furniture is in the room, where you are in a series of rooms, where you are in a particular space. Can you hear the audio on this video that I'm playing right now?
I can kind of. It sounds very.
Those bits according to simple rules. Computers encode information as a series of bits.
And in this video, what we're seeing, for those who are just on the audio, is that it's a video of a 3D modeled room, and there's it's a perspective of walking through a room,
and then manipulate
down a hallway
according to simple rules,
and walking to another room closer to where a speaker is,
of bits,
the source
which can be either 0 or 1, and then manipulate.
So you get the idea that the sound is enabling the artificial intelligence to reconstruct the space accurately. And it's not just accurate within one space. It can be accurate to bouncing sound around walls and through doorways and back and forth over furniture. And suddenly we're starting to see a very interesting, what I would imagine is how a bat or a dolphin or animals who use sonar to navigate their environments, how they may be able to experience the environment through sound.
I think the tough thing about them is that they can also see. So they have even for bats, if it's dark out, they still have some limited vision, and right. So they have some information that they can add to that to help themselves out. That video you were showing, Kiki, that was the image that the AI constructed.
Yes.
So how did the AI know how to color the legs of the chairs different from the padding?
That's probably been constructed by the...
Great. I was just curious if they fed some interior design information into the AI as well, because they could, right? Like looking at what's happening with Midjourney and stuff like that, you could totally feed
information.
Interior design information into them, and they could figure out probability of like, okay, this shaped thing is probably a chair. And it's probably a fabric chair based on the way that the sound is doing that. And they would know that because those are the shapes chairs can make, right?
So was it 6-8 years ago? that the a group out of MIT hacked a printer, I think it was, the little light that's on a printer, to be able to detect sound in a room. Some really bizarre thing like that. So now you're talking about if you could hack a printer to get the layout of a room by using that light to sense the sound in the. Wow.
Right. So now this is starting to combine these things where we're starting to get the combination of the construction of spaces and also the experience of the sound of the user. So if you are a virtual user within a virtual space, right? You're going into some VR space or into a game or something. It's going to give you more accurate as it creates the space. It's going to give you more accurate sound based on where the source of the sound is and where you are situated with respect to that sound. So it's not new. It's not like all new stuff. This is another iteration of all of this work coming together. Yeah. Anyway, it's more accurate. The sound models are better. The geometry is better. The model's able to be able to apply the acoustic information better. Anyway, yeah, MIT, it's reinventing sonar. I mean, I was kind of getting at this has been done already by nature.
The last
now we're doing it for AI.
Last thing I'll tag onto this is that you could absolutely use this then for mapping underground caverns. I mean, you can put in anywhere you could put a microphone. I guess at this point you could put a camera with a light anyway to look around.
Right, but mapping exact distances with a camera is a lot harder than it could be with this. Yeah, I was thinking the same thing.
Drop it down into an underground cavern or a collapsed building or whatever, and see where the pockets are. There's some pretty interesting uses for this. And you need some smart people to sit down and come up with a bunch of cool things they could do with this technology if they had it.
I need the mapping of the caverns, and then I can go in virtual reality and explore the caverns and find a really cool cavern where I can have that piano concert.
There you go. Yeah.
Have we done it? Have we made it to the end of the show?
We have.
We did it, everyone. We vomited out. We picked out all the best science for you. I'm glad you ate it up. Anyway, thanks for listening, everyone. Thanks for joining us. We do always enjoy that you come here to enjoy the science with us. Shout outs to a few people. Fada, who's not here tonight because he's off doing improv comedy. Learning some improv, which is very cool. But Vada, thanks so much for your help on social media and also with show notes. Gord and Aaron, Lauren, others, thank you for keeping our chat rooms happy, nice places to be. Identity 4, thank you for recording the show. And Rachel, thank you for your editing and help in publishing the show. And of course, I cannot forget all of our Patreon sponsors. I definitely need to take this opportunity to read out a few names. Thank you to Teresa Smith, James Schaefer, Richard Badge, Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tozzi, Woody MS, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Gaurav Sharma, Reagan, Derek Schmidt, Don Mundis, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus 104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, David Youngblood, Matt Bass, Vote Beto for Texas, John McKee, Greg Rayleigh, Mark Hessenflow, Steve Leisman, a.k.a. Z. McKenhays, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Remy Day, Flying Out, Christopher Dreyer, Greg Briggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rondi Lewis, Paul, Rick Rimmers, Pilt Shane, Kurt Larson, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kedem Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, and Jason Roberts. Thank you. Thank you all for supporting TWIS on Patreon. And if you're interested in supporting us on Patreon, head on over to twis.org and click on that Patreon link. On next week's show,
We will be at episode 900
Mm-hmm.
of the podcast version of the show.
Yeah.
And we will be back Wednesday, 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels, as well as from twis.org/live.
Hey, do you want to listen to us as a podcast? Perhaps while you roll some wooden beads around for fun. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up for a newsletter.
You can also contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will get gobbled up, and we will never be able to vomit it back out again because we will have taken some new experimental drugs.
Woot!
If
Sounds
you're tired
exciting.
of hearing about vomit, let us know on Twitter, where we are at TwistScience, at Dr. Kiki,
We won't
at
do
Jackson
anything
Play,
about
and
it.
Blair's Menagerie. We love your feedback. If there's a topic you'd like to have us cover or address, suggestion for an interview, a haiku that comes to you in the night, please let us know.
Listen, I love your feedback, but if you tell me to stop talking about snot or vomit, you will be ignored. Just FYI. All right, we'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, you're supposed to remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.